Chapter 2: Synapses. If you had to communicate with someone without sight or sound, what would you do? Chances are your first choice would be a touch code or a system of electrical impulses. You might not even think about chemicals passing back and forth. Chemicals are, however, the main way that your neurons communicate. They communicate by transmitting chemicals at specialized junctions called synapses. The concept of the synapse. In the late 1800s, Ramon y Cajal anatomically demonstrated a narrow gap separating one neuron from another. In 1906, Charles Scott Sherrington physiologically demonstrated that communication between one neuron and the next differs from communication along a single axon. He inferred a special gap between the neurons and introduced the term synapse to describe it. Cajal and Sherrington are regarded as the great pioneers of modern neuroscience, and the nearly simultaneous discoveries supported each other. If communication between neurons is special in some way, then there can be no doubt that neurons are anatomically separate from one another. Sherrington's discovery was an amazing feat for scientific reasoning, as he used behavioral observations to infer the major properties of synapses half a century before researchers had the technology to measure those properties directly. Properties of the synapse. Sherrington studied reflexes, automatic muscular responses to stimuli. In a leg flexation reflex, a sensory neuron excites a second neuron, which in turn excites a motor neuron, which excites a muscle. The circuit from sensory neuron to muscle response is called a reflex arc. If one neuron is separated from another, as Cajal had demonstrated, a reflex must require communication between neurons, and therefore measurement of reflexes might reveal some special properties of that communication. Sherrington strapped a dog into a harness above the ground and pinched one of the dog's feet. After a fraction of a second, the dog flexed, raised its pinched leg, and extended its other legs. Sherrington found the same reflexive movements after he made a cut that disconnected the spinal cord from the brain. Evidently, the spinal cord controlled the flexation and extension reflexes. In fact, the movements were more consistent after he separated the spinal cord from the brain. In an intact animal, messages descending from the brain modify the reflexes, making them stronger sometimes and weaker at others. Sherrington observed several properties of the reflexes that suggest that special processes at the junction between neurons is 1. Reflexes are slower than conduction along an axon. 2. Several weak stimuli presented at nearby places or at times produce a stronger reflex than one stimulus alone does. And 3. When the set of muscles becomes excited, a different set becomes relaxed. Let's consider each of these points and the implications. Speed of a reflex and delayed transmission at the synapse. When Sherrington pinched a dog's foot, the dog flexed that leg after a short delay. During that delay, an impulse had to travel up an axon from the skin receptor to the spinal cord, and then an impulse had to travel from the spinal cord back down the leg to a muscle. Sherrington measured the total distance that the impulse travelled from skin receptor to spinal cord to muscle and calculated the speed at which the impulse travelled to produce the response. He had found that the speed of conduction through the reflexes arc varied, but was never more than about 15 meters per second. In contrast, previous research had measured the action potential velocities along a sensory or motor neuron nerves at about 40 meters per second. Sherrington concluded that some process must be slowing the conduction through the reflex, and he inferred that the delay occurs where one neuron communicates with another. This idea is critical, as it established the existence of synapses. Sherrington, in fact, introduced the term synapse. Temporal summation. Sherrington found that repeated stimuli within a brief time had a cumulative effect. He referred to this phenomenon as temporal summation, meaning a summation over time. A light pinch of a dog's foot did not evoke a reflex, but a few rapidly repeated pinches did. Sherrington surmised that a single pinch did not reach the threshold of excitation of the next neuron. The neuron that delivers the transmission is the presynaptic neuron, and the one that receives it is the postsynaptic neuron. Sherrington proposed that although the subthreshold excitation in the postsynaptic neuron decays over time, it can combine with secondary excitation that follows it quickly. With a rapid succession of pinches, each pinch adds its effect to what remained from the previous ones until the combination exceeds the threshold of the postsynaptic neuron, producing an action potential. Decades later, Sherrington's former student, John Eccles, attached microelectrodes to stimulate axons of presynaptic neurons while he recorded from the postsynaptic neuron. For example, after he'd briefly stimulated an axon, Eccles recorded a slight depolarization of the membrane of the postsynaptic cell. Note that this partial depolarization is a graded potential. Unlike action potentials, which are always depolarizations, graded potentials may either be depolarization, excitatory, or hyperpolarization, inhibitory. A graded depolarization is known as an excitatory postsynaptic potential, and it results from a flow of sodium ions into the neuron. If an excitatory postsynaptic potential, EPSP, does not cause the cell to reach its threshold, the depolarization decays quickly. When Eccles stimulated an axon twice, he recorded 2 EPSPs. If the delay between the EPSPs was short enough, the second EPSP added to what was left of the first one, producing temporal summation. At point 3 in figure 3.3, a quick sequence of EPSPs combines to exceed the threshold and produce an action potential. Spatial summation. Sherrington also found that the synapses have the property of spatial summation, that is, summation over space. Synaptic inputs from separate locations combine their effects on a neuron. Sherrington again began with a pinch too weak to elicit a reflex. This time, instead of pinching 1 point twice, he pinched 2 points at once. Although neither pinch alone produced a reflex, together they did. Sherrington concluded that pinching 2 points activated separate sensory neurons, which axons converged into one neuron in the spinal cord. Excitation from either sensory axon excited that spinal neuron, but did not have enough to reach the threshold. A combination of excitations exceeded the threshold and produced an action potential. Again, Eccles confirmed Sherrington's inference, demonstrating that EPSPs from several axons summate their effects on a postsynaptic cell. Spatial summation is critical to brain functioning. In most cases, sensory input at a single synapse produces only a weak effect. However, if a neuron receives many incoming axons with synchronized input, spatial summation excites the neuron enough to activate it. Temporal summation and spatial summation ordinarily occur together. That is, a neuron might receive input from several axons in close succession. Integrating these inputs provides complexity. A series of axons activate in one order can have different results from the same axons in a different order. For example, a neuron in the visual system could respond to light moving in one direction and not another. Inhibitory synapses. When Sherrington vigorously pinched a dog's foot, the flexor muscles of that leg contracted, and so did the extensor muscles of the other 3 legs. You can see how this arrangement would be useful. A dog raising one leg needs to extend the other legs to maintain balance. At the same time, the dog relaxed the extensor muscles of the stimulated leg and the flexor muscles of the other legs. Sherrington's explanation assumes certain connections in the spinal cord. A pinch on one foot sends a message along a sensory neuron to an interneuron, and that excites the motor neurons that connect to the flexor muscles of that leg and the extensor muscles of the other legs. Also, the interneuron sends messages to inhibit the extensor muscles in that leg and the flexor muscles of the other 3 legs. Later researchers physiologically demonstrated the inhibitory synapses that Sherrington had inferred. At these synapses, input from an axon hyperpolarizes the postsynaptic cell. That is, it increases the negative charge within the cell, moving it farther from the threshold and decreasing the probability of an action potential. This temporary hyperpolarization of the membrane, called an inhibitory postsynaptic potential, IPSP, resembles an EPSP. An IPSP occurs when synaptic input selectively opens the gates for potassium ions to leave the cell, carrying a positive charge with them, or for chloride ions to enter the cell carrying a negative charge. Today we take for granted the concept of inhibition, but at Sherrington's time the idea was controversial, as no one could imagine a mechanism to accomplish it. Establishing the idea of inhibition was critical not just for neuroscience, but for psychology as well. Whenever we talk about inhibiting an impulse, we use the concept first demonstrated by Sherrington. Relationship among EPSPs, IPSPs, and action potentials. Sherrington's work opened the way for exploring wiring diagram of the nervous system. Consider the neurons shown in figure 2.8. When neuron 1 excites neuron 3, it also excites neuron 2, which inhibits neuron 3. The excitatory messages reach neuron 3 faster because it goes through just one synapse instead of two. The result is a burst of excitation, EPSP, in neuron 3, which quickly slows or stops. You see how inhibitory messages can regulate the timing of activity. To see how the wiring diagram in the nervous system controls the outcome, consider figure 2.9. The axon from either cell A or cell B stimulates cell X with +1 unit. If the threshold of cell X is +1, then the X cell responds to A or B. If the threshold is +2, then the X cell responds to A and B. With little effort, you can imagine other constructions. Many mathematical models of the nervous system are based on the connection like these. However, researchers have discovered complexities that Sherrington did not anticipate. Some synapses produce fast, brief effects, and others produce slow, long-lasting effects. In many cases, the effect of two synapses at the same time can be more than double the effect of either one, or less than double. Certain combinations of synapses summate with one other more strongly than others do. Also, the strength of a synapse can vary from one time to another. The nervous system is indeed complex. Most neurons have a spontaneous firing rate, a periodic production of action potentials even without synaptic input. In such cases, the EPSPs increase the frequency of action potentials above the spontaneous rate, whereas in IPSPs decrease it. For example, if the neuron's spontaneous firing rate is 10 action potentials per second, a stream of EPSPs might increase the rate to 15 or more, whereas a preponderance of IPSPs might decrease it to 5 or fewer. Module 2.1, in closing, the neuron as a decision maker. Transmission along an axon merely sends information from one place to another. Synapses determine whether to send the message. The IPSI and the IPSPs and the EPSPs reaching a neuron at a given moment compete with one another, and the net result is complicated, not exactly the algebraic summation of their effects. We could regard the summation of EPSPs and IPSPs as a decision, because it determines whether or not the postsynaptic cell fires an action potential. However, do not imagine that any single neuron decides what you will eat for breakfast. Complex behaviors depend on contributions from a huge network of neurons. Summary. 1. The synapse is the point of communication between two neurons. Charles S. Sherrington's observation of the reflexes enabled him to infer the existence of synapses and many of their properties. 2. Because of transmission for a reflex arc is slower than transmission for equivalent length of axon, Sherrington concluded that some processes at the synapses delays transmission. 3. Graded potential EPSPs and IPSPs summate their effects. The summation of graded potentials from stimuli at different times is temporal summation. The summation of potentials from different locations is spatial summation. 4. Inhibition is more than just the absence of excitation. It is the active brake that suppresses excitation. For effective functioning of the nervous system, inhibition is just as important as excitation. 5. Stimulation at a synapse produces a brief graded potential in the postsynaptic cell. An excitatory graded potential depolarization is a EPSP. An inhibitory graded potential hyperpolarization is a IPSP. An EPSP occurs when the gates open to allow sodium to enter the neuron's membrane. An IPSP occurs when the gates open to allow potassium to leave or chloride to enter. Number 6. The EPSPs on a neuron compete with the IPSPs. The balance between the two increases or decreases the neuron's frequency of action potentials. Thank you. This is the end of section 2.1.